In terms of RNA-seq, you'll notice RNA-seq over here at the bottom right. This is the newest area of functionality in SVS. And what we're supporting at the moment is differential expression analysis. And similar to the DNA sequencing tools, there are a few limitations to be aware of. In DNA, we don't do alignment or variant calling. We expect that to be already done. That's why we were starting from VCF files and using BAMs just for visualization. With RNA, it's the same way. We don't do alignment, we don't do quantification, we don't do novel transcript discovery or isoform discovery. We expect you've already done that, and what you're coming to us with, whoops, that's not the project I wanted to open, is something where you already have that quantification done. And this is an example where there are 15 samples representing 3 different types of breast cancer tumors, and there's a column for every gene together with the observed quantifications for that gene. And the input format is described in the tutorial for RNA-seq. We hope to be able to support some additional input formats soon. We'll also be adding some more functionality. But what I want to show you here is a few simple workflows for doing some data cleanup, normalization, and analysis for differential expression. So starting out here, we see this spreadsheet where we have a lot of various values. Some genes you can see are very poorly expressed, some are very highly expressed, and depending on your workflow, you might be really interested in the low expression genes, you might not be. What I'd like to do as a starting point is require that within my 3 sample groups, so I've got luminal, basal, and claudin type, high claudin type breast cancers, I want at least 2 samples in each group to have some minimal amount of expression to be considered. So in the RNA-seq menu, well, first I'll set that as my dependent variable by clicking on it, just like we saw with the CNV and GWAS tools. Then I have a neat little script here that I can set up to require at least 2 occurrences or 2 samples within the dependent categories to have, I'll just say, at least a 1 read expression level. And you could set that up a lot of different ways. Again, I'm just demonstrating functions today more than best practice workflows. Then I'll subset this. So at this point, maybe I want to get an idea of what the expression looks like in these samples, whether they have similar distributions or not. And I have a little script here that's going to produce 15 box plots. And as I look at this, all I'm seeing is outliers. So the majority of the data, the median and the quartiles, is just completely smashed down here at the bottom. Obviously, I need to do some kind of normalization on this spreadsheet. So here in the RNA-seq menu again, I have a few different options for normalizations. I'm just going to run the default, which is to do an upper quartile normalization and log2 transform. And so it gives me both of the intermediate outputs, the upper quartile and the log2 of that. And I'll repeat my little box plot procedure just to see if this cleaned things up. And in fact, it did. We've gotten rid of all of those high outliers. We've brought everything back into a range where the samples can be compared more directly. It was an upper quartile normalization, so you can see that the 3rd quartile is the same across all of these. And so with that normalization done, I can do other kinds of analysis on this data. If I wanted to run a t-test or a ANOVA test to look at differences in expression levels here, I could do that. I also have the option of looking at something like principal components analysis, which we've already seen with SNPs, but we haven't seen it done with numbers. I'll go ahead and let this run. So here now, just like we saw with the SNPs earlier, we've got a general idea of what the expression patterns, or not the expression patterns, what the principal components are. And I'm just going to merge that onto the phenotypes like we've seen before so that I can visualize these. Maybe look at the first and second components, recolor these by breast cancer type, and now I see it looks like we have some visually obvious clustering. Let me find the button I'm looking for here. That's the one. Between the three types. So the basal, luminal, and high Claudin types are all clustering together. That's a strong indication that we'll find some kind of genetic explanation for the or some pattern in the expression that explains the difference between the tumor types. So now if I want to run an association test, I could use that same normalized transformed data as the basis for an association test using, again, the t-test or regression or ANOVA type tools. But what I'd like to use here is come back to the data prior to the normalization being applied and use the DE-seq method. So DE-seq is a bit more sophisticated method for association with RNA-seq. And I will compare luminal versus basal. So it is designed just to do pairwise comparison between two groups. There are some options for extra outputs. I will turn that on. I'll go ahead and let this run. Now, the reason I went back to the pre-normalized data is because DE-seq does its own normalization process. If you look at the online tutorial for RNA-seq analysis, it gives a few notes about different input types when you do and don't want to use different normalizations. There's really not a lot to it there, but depending on whether you're coming from RSEM or cufflinks, and if you have FPKMs or something else, you may want to use some different options. And again, that's laid out in the tutorials. But after running this, the main output we get here has p-values per gene. I think it also has fold change information, some other statistics related to each gene and the various sample groups there. Now, if you want to sort on p-value, for example, you can see LEPRL1 comes to the top as the gene with the strongest statistical significance. If you want to look at this in terms of a volcano plot, where you have log fold change versus log p-value, here now we can click around and explore and see what gene this is. As expected, LEPRL1 is the one that has that most significant p-value. But we can also look at it in terms of the effect size down here with the fold change. And as we click around, we see what gene is represented in each place. Now, what I really like to do is take these p-values and throw them into the browser. And we've seen this before. This should be a familiar environment by now. This is probably LEPRL1 right over here, and indeed it is. As I zoom in, it's being labeled. But what is really fun now is to add in a heat map of the normalized intensities, or the normalized expression values. And finally, to group these based on type. We're just comparing luminal and basal, so I'll hide the Claudins. So this blue axis bar is the luminal, the orange is the basal, and you can see low expression in one group, high expression in another. It's quite a clear pattern, and you can further to this, if you need to explore the genes a little bit, you can add in annotation data about the genes, whether that be gene summaries from RefSeq or from OMIM, or I like the DBNSFP summaries right here. You can add those all in. So in this case, I can look at the DBNSFP summary and see little bit about the gene. It's related to myopia. That's interesting. I was expecting something for breast cancer, but it is expressed in a lot of different places. If I brought in the cosmic track, I believe this one is pretty well annotated for breast cancer. If we look at right here, cosmic, look at that link. Anyway, with that, I think we're at the place now that we can start to wrap this up. As we've gone through everything today, we have shown you a broad, oh, actually there's one more thing I wanted to show. And we looked at the genome browser-based heat map. We can also do heat maps just based on the most significant genes. We can do this clustered heat map view with the dendrograms, so you can see the relationships with the hierarchical clustering between both samples and genes.